Welcome back. We're into section 2. This is going to have two videos. The first one we'll look at moving proteins into the endoplasmic reticulum, because we talked about the endomembrane system and sorting through the system and secretion, but we didn't really address how proteins get into the endomembrane system. So we'll look at how proteins get into the ER after we've done a quick introduction to the phenomena of protein sorting. And in the second video, in this 19.5 section, we'll take a look at how we get proteins into the mitochondria and the chloroplast, for example. So let's get going. Let's get the right slide up here. There we go. Okay, so thinking about protein sorting from kind of like a 10,000 foot perspective, if you like. Think about any protein that is synthesized. It has to be synthesized, post-translationally modified, and sorted and directed to its final location where it's going to have its function in the cell. And so we can think about kind of like three main categories of places where proteins will end up in the cell. So the first one is the endomembrane system. We talked about that quite extensively a little while ago. Then we can think about, I guess, the cytosol. That's another major compartment. And then we could think about the mitochondria, the chloroplast, the peroxisomes, and the interior of the nucleus, all as another set of places where cells have to be directed to. So most translation initiation, in fact all translation initiation, except unless we're talking about translation in the mitochondria and the chloroplast, initiates in the cytosol. And then there are kind of, once it initiates here, the kind of two paths diverge about the fate of a protein that's being translated. This kind of first pathway over here is what we'll deal with in this video, and that is getting proteins into the endomembrane system, ultimately thinking about either targeting to the plasma membrane, the lysosome, or for secretion, function in the Golgi, that kind of thing. The other pathway is the pathway which deals with targeting to the mitochondria, the chloroplast, peroxisomes, and the interior of the nucleus. So once translation initiates here, then proteins have these short targeting sequences, and that targeting sequence will tell the cell this protein needs to go to one of the 3 or 4 compartments in the mitochondria, so the matrix, the intermembrane space, or the inner or outer membrane, if we think about those as compartments. They're not compartments, they're just structures. Same thing for the chloroplast. The targeting sequence has got to send the protein either to the outer or inner membrane, or to the stroma, or into the thylakoid, or even into the thylakoid lumen, even to the thylakoid membrane, I guess there. So 5 places. Then we can think about targeting the peroxisome, and then we're going to think about how you get molecules into the nucleus. We talked about nuclear import in a previous video, so we won't get into that into too much detail. So think about this generally in terms of 2 major pathways after you initiate translation. One is into the endomembrane system, and the other is to pretty much everywhere else. So we'll do the pretty much everywhere else after we've done the endomembrane system targeting in this video. So that first kind of pathway is utilized by ribosomes synthesizing polypeptides that are destined for function in the ER, function in the Golgi, function in the Golgi membrane, function in the plasma membrane, function in the lysosomes, or function outside the cell to be secreted. And so those cells that initiate translation in the cytoplasm ultimately become attached to the endoplasmic reticulums early in translation. So even though translation initiates in the cytosol, very quickly that ribosome translocates to the ER membrane, and that's where translation continues. So it's a pretty short amount of translation that occurs in the cytosol before the ribosome goes to complete translation at the ER membrane. And so once the ribosome that initiates translation in the cytosol, once that translocates to the ribosome, the rest of translation occurs through this kind of channel called a translocon in the ER membrane, and this is called co-translational import. So the membrane, the ER protein is translated through this protein pore in the ER lumen, in the ER membrane, and into the ER lumen. So that's called co-translational import. The other form of import is post-translational import. And so that alternative mechanism of post-translational import is for polypeptides that are going to go to the mitochondria, the chloroplast, nuclear import, peroxisomes, those kinds of things. And so there is what happens is after translation is complete, the protein is processed in the cytosol and then targeted to the appropriate organelle. So that's called post-translational import. So we've got co-translational import, which happens on the ER membrane to get proteins into the endomembrane system, and then we've got post-translational import, which is to get proteins into places like the nuclear inside of the nucleus, the nuclear matrix, or into the mitochondria, or into the chloroplast. So let's deal with the endomembrane system here, and then we'll do another video and we'll talk about post-translational import. So a couple of guys, Günter Blobel, the only reason I tell you his name is he's got a funny name, Günter Blobel, and David Sabatini were the first people to suggest what we call the signal hypothesis. And this suggests or proposes that proteins move into the ER during synthesis because they have some kind of intrinsic signal directing them to the ER and into the ER lumen. And so the ER signal sequence, as it's called, directs the mRNA polypeptide complex to the rough ER surface. So we have an mRNA being translated on a ribosome, producing a short piece of polypeptide, and that short piece of polypeptide is turns out to be a 20 amino acid sequence. It's discovered by a guy called Milstein. 20 amino acid sequence on the N-terminus, which is responsible for getting that polypeptide into the ER. And this was discovered with the light chain of immunoglobulins, obviously a secreted protein, so that goes into the ER lumen to be processed through the endomembrane system and be secreted. So how did that all get found out? So there's a couple of diagrams here we'll look at. So this top one here, you can perform translation in a cell-free system. So you can make an extract of cells, and actually the cells that were used to do this initially were rabbit reticulocytes. So basically red blood cells from exsanguinated rabbits, and so when you get that cell-free extract, you get all the machinery required for translation. And so that would be a cell-free system because there's no cell. You just take the cytoplasm and you actually get rid of any other organelles and just have this cell-free system. Now, to that system, you can add ER membrane fragments, and they form vesicles called microsomes. So this is cell-free translation up here, microsome-free. And so we've got translation occurring and a mature protein being formed. And then down here we've got fragments of ER membrane that have been added and microsomes have formed. So absent the microsomes in the top here, you get this mature protein with this short 20 amino acid segment on the N-terminus. But if you add microsomes, is what you find there is that translation occurs into the kind of lumen of these microsomes, and mature proteins lack that 20 amino acid signal. And so subsequently, if you delete the coding sequence for this 20 amino acid sequence from the gene for example, the light chain of immunoglobulin, you don't get insertion into the ER lumen. And so this kind of demonstrates that there's this 20 amino acid signal, and that's the signal sequence which sends some proteins to the ER lumen. And so those proteins that get sent into the ER lumen and then have to be further processed by having the signal sequence taken off are called preproteins. That signal sequence is about 15 to 30 amino acids, averaging about 20. It's kind of usually positively charged towards the N-terminus, and then there's a short central hydrophobic part, and then a polar part where the cleavage from the mature protein takes part. So it's only 20 amino acids long, but it's got some very discrete characteristics. And the good thing about that is you can make a good attempt to identify them using bioinformatics, looking at the chemical characteristics of the amino acids. So how do we actually get these proteins destined to the ER lumen into the ER lumen using this signal sequence? So the interaction between the growing polypeptide that's being translated and the endoplasmic reticulum is under the control of this protein here called SRP. That's the signal recognition particle, and it's recognizing the signal sequence, obviously. So let's kind of and so SRP basically binds to the signal sequence and drags the ribosome to the ER membrane surface, where you then get co-translational import. So let's kind of take a look at this in some steps. So first two steps here. Once the ER signal sequence is translated there, translation pauses. This sequence causes translation to pause briefly, and the SRP binds to the signal sequence in the cytosol, and that causes then the whole ribosome polypeptide mRNA complex to dock on the SRP receptor, which has got two subunits there. So that's steps 1 and 2. In step 3 there, the SRP has delivered the kind of nascent, the growing polypeptide to the SRP receptor. So we're here, so we've got docking occurred here. And then that interaction is reinforced by the binding of GTP to SRP and GTP to the alpha subunit of the SRP receptor. So this kind of locks everything in place. Then we have transfer of the nascent or the growing polypeptide into a pore in the membrane, and so this purple protein here is called the translocon. And so when the ribosome docks, the translocon is closed, and so once you initiate translocation of the polypeptide here, the translocon opens. And then you get threading of the growing polypeptide through the channel in the ER lumen made by these proteins. And at that point you have hydrolysis of GTP to GDP and organic phosphate by SRP in the alpha subunit of the SRP receptor. And that releases SRP to go off and do its own thing. We continue translation through the translocon, and a protein called signal peptidase cuts off the signal sequence. So that signal sequence won't be present in the full length protein. So that's an example of that processing that we talked about, where you might cut off the N-terminus of the protein. And so we cut off the signal sequence using signal peptidase. We keep threading the growing polypeptide chain through, and eventually the whole chain is released into the ER lumen, and it will be folded and checked for folding using some of the mechanisms we've discussed in an earlier video. So that's how we get a folded soluble protein into the ER lumen. But we also have to think about how we get in integral membrane proteins. So this can be a membrane protein that's going to be integral in the ER membrane, or in the Golgi, or in a lysosome membrane. So for example, the ATPases, the proton pumps, the ATPases that acidify the lysosomes, they've got to be gotten into those membranes somehow. What about all those proteins in the cell membrane that are transmembrane? So when you look at transmembrane proteins, there are 5 or 6 different groups we can look at. Type 1 here, don't worry about all the examples, we're just worried about the types. Type 1 here is a carboxy terminus that would be in the cytosol. So that's the cytosol of the cell, obviously. A transmembrane segment, a single transmembrane segment, and then an amino terminal domain, which would be in the exoplasmic space. So that could be inside the ER or the Golgi, or inside the cell or the cell exterior. So that would be outside the cell in that case. Type 2 integral membrane proteins are the same, but they're reversed. So amino terminus outside, in the cytosol, and then the carboxy terminus exoplasmic. Type 3 is a kind of variation of type 1. Type 4 is kind of a variation of type 2, and then type 3 with these kind of 2 variants, and type 4 are these more complicated proteins which have multiple hydrophobic alpha helices, so they're multi-pass transmembrane proteins. And so 1, 2, 3, 4, 5, 6, 7, this would be something like a G protein coupled receptor involved in cell signaling. And so the translocation process to get these hydrophobic regions into the membrane, you can imagine, becomes quite complicated. So we're not going to look at this. We'll focus on the type 1, type 2, and type 3 insertions. So let's take a look at how you get type 1 single pass integral membrane protein. So what we've got to do is focus down here at the end to start with. So what we're going to do is we've got to get a protein with a single transmembrane region and a carboxy terminal region, which is in the cytosol, and then in the ER lumen we're going to get the amino region, so the amino terminus of the protein. So you can think about whether this amino terminus would be inside the Golgi, in the lumen of the Golgi, whether it would be outside of the cell or inside of the cell. That's kind of the kind of thing that I'll be asking you on some of your quizzes. So, how do we go from starting translation initiation up here on the translocon to ending up with this process down here? There are a couple of mechanisms that people think this works by, and I'm just going to tell you one mechanism, one model. And so the mechanism that we're going to talk about here is where we have a protein which has a N-terminal signal sequence. So that was like what we just saw for getting a soluble protein into the ER lumen. So the whole kind of docking and translocation initiation is the same as for a soluble ER lumen protein. So this involves SLP and the SLP receptor and signal peptidase. And so as what's going to happen here is we're going to have translocation, cleavage of the signal sequence by signal peptidase over here on the left, and then we're going to have continuation of translation and insertion of the amino terminus into the ER lumen, and then at some point, some way down the protein, there's a sequence of amino acids called the stop transfer anchor sequence. And so this sequence of amino acids here is going to form the hydrophobic transmembrane segment. So when this region of the amino acids, or the sequence of amino acids, gets translated, the ribosome pauses, and the rest of the polypeptide is synthesized to the outside. And so you can see there's a slight release of the ribosome at this point here, and that means that the translocon is going to close, and the rest of the translation of the protein will be on the outside and the cytosolic side of the ER membrane. And so what we end up with is we complete translation. We end up with a cytosolic carboxyl domain and a ER lumen amino terminal domain. And then whenever we move that through the endomembrane system, we're always going to maintain that kind of sidedness, if you like, with the amino terminus inside whatever structure we're looking at, and the carboxy terminus in the cytosol. So if you think about what happens when a vesicle, for example, containing this membrane protein fuses with the cell membrane, you can work out whether the carboxy terminus would be outside the cell or inside the cell, and the same for the amino terminus. So what happens at this point here in step 4 is as the kind of ribosome is starting translation outside of the ER lumen, this hydrophobic segment here kind of gets dragged out of the translocon into the membrane, and that's when it becomes the transmembrane segment. Okay, let's look at type 2 single passes. So after the internal signal anchor sequence here, so the mechanism for type 2 is similar to type 1. We've got an internal signal anchor sequence, but no signal sequence on the amino terminus, and so we translate we do a translation all the way to the point where we have the internal signal anchor sequence. So after the internal signal anchor sequence is synthesized on the cytosolic ribosome, that's when we get SLP binding, which directs the ribosome to the ER membrane. So that's again similar to the targeting of the soluble proteins that would be secreted or that kind of thing. Except that the kind of the hydrophobic signal sequence here is located kind of inside the polypeptide chain rather than on the N-terminus. And so the nascent chain becomes oriented in the translocon with its N-terminus sequence on the cytosolic face. And then as we continue translation through the translocon, we get the carboxy terminus ends up on the inside, and we have this mechanism where the transmembrane segment is pulled out of the translocon and into the membrane. And a lot of this is determined by this run of positively charged amino acids here interacting with the ER lumen surface there. So that's type 2. Type 3, very similar to what you see for type 2, except that the positively charged amino acids are on the C-terminal side of the signal anchor sequence. So if we back up, you see here this run of 3 amino acids, the run of charges, positively charged amino acids here is on the amino side of the polypeptide chain. But when we're looking at type 3 single pass polypeptides, then the run of charges on the polypeptide chain, so the positively charged amino acids, is on the C-terminal side of the anchor stop sequence. And so that's the only real difference here in terms of inserting a type 3 single pass integral membrane protein. And so that causes the amino terminus to be inside. We pause when we translate the anchor stop signal sequence, and then the run of positively charged amino acids causes the C-terminus of the protein to end up being translated on the cytosolic side. So this gives us kind of the backwards version of what you saw for type 2. So it's only due to this it's due to this difference in the positively charged amino acids that you see there. So that's how you get soluble proteins into the ER lumen, or into the Golgi interior ultimately, or secreted from cells, and then how you deal with getting type 1, type 2, and type 3 integral membrane proteins into the ER membrane, and then they may become membrane proteins in the Golgi, or in lysosomes, or in the cell membrane. So that's it. I'll be back soon.